of this program, and that's Dr. Glenda Courtney-Martin. Glenda is a health clinical scientist and senior associate scientist at SickKids Hospital. She also carries affiliation as an associate professor in the Department of Nutritional Sciences at U of T. Her research centers on the protein requirements and quality of foods for human nutrition using metabolism-driven amino acid-based methods. Today, she will present a snapshot of that work using the concepts and application of a stable isotope-based indicator amino acid oxidation method. Glenda, the floor is yours.
Okay, says someone else is sharing. Okay, I'll share.
Just have to put that into presentation mode. There you go.
Okay, so thank you very much for the invitation. Very happy to be here today. So I'm going to present, and the topic of my presentation is From Amino Acid and Protein Requirements to Protein Quality, Knowledge Gained from Applying this Stable Isotope-Based Indicator Amino Acid Oxidation Method. So these are my declaration of interest. My research program is focused, as Andrew said, on protein and amino acid requirements and metabolism and on protein quality. And my research is in neonates, looking at amino acid requirements in preterm neonates on total parenteral nutrition in the NICU to older adults over 60 years. And all of my research uses stable isotopes. We use C13-phenylalanine and 13C sodium bicarb. And for our protein and amino acid requirement studies, and N15 glycine and an N+3 glycine for other metabolic studies. And so the objectives today is to present some background on protein and amino acid requirements and to present some concepts and application of the indicator amino acid oxidation method. I'll also present a couple physiological studies looking and comparing the IAAO-derived protein requirements and also to look at the application to study amino acid requirements, particularly in the elderly, and hopefully I get to some protein quality work. So as we know, proteins is an essential component of the diet, but amino acids are the currency of protein metabolism. And so we eat proteins in order to gain the 20 amino acids, particularly the 9 essential amino acids that are required. And it's important that a protein requirement should be defined not only quantitatively, but qualitatively to meet the requirements of the 9 essential amino acids. And so current protein recommendations, protein and amino acid recommendations are set in North America by the Institute of Medicine, and they come under the umbrella of the Dietary Reference Intakes, the DRIs, and for the world by the Food and Agricultural Organization of the United Nations, the FAO. And these recommendations are largely for adults. They're based on data derived from young adults only, particularly young adult males. The protein requirements in particular is set based on nitrogen balance data, whereas the amino acid recommendations are based on carbon oxidation studies from our lab and the lab of Anura Kirpad in India. And so the current protein requirement is based on a meta-analysis from Rand et al. Of the world nitrogen balance data using linear regression analysis. And so the recommendations are set at 0.6 grams per kilo per day, which is the estimated average requirements, and 0.8 grams per kilo per day, which is the recommended dietary allowance. Now, nitrogen balance is a pretty elegant method, but its application is pretty challenging. And so the DRI committee at the time, who set the recommendations for protein, concluded that due to the shortcomings of the nitrogen balance method, it is recommended that the use of nitrogen balance no longer be regarded as the gold standard for the assessment of the adequacy of the protein intake and that alternative methods should be sought. And so our group decided to apply the indicator amino acid oxidation method to study protein requirements. And so the IAAO method is based on the concept, it's a physiological premise that amino acids are not stored. And so indispensable amino acids in particular are either incorporated into protein and non-protein substances or are oxidized. And so when you feed a test and a limiting amino acid in graded intakes or a total protein in graded intakes, we use an indicator amino acid, and that is C13-phenylalanine, which is most commonly used by our group, to assess the oxidation of that indispensable amino acid in response to variations in protein intake or variations in amino acid intake. And so as we increase the intake of the test or amino acid or the test protein, indicator amino acid oxidation decreases. And when we get to a point where the indicator amino acid oxidation no longer decreases, that is the mean requirement. And so further increases in the protein or the amino acid intake produces no further changes in the oxidation of the indicator amino acid. Now, this was validated by Ball and Bailey at the Guelph University in pigs many years ago in 1986. And what they saw was that indicator oxidation is the inverse of protein synthesis. And they did this by looking at retention of C14 labeled phenylalanine in liver tissue of pigs. And what they found was that as they increased the intake of protein or amino acid, the retention of the indicator, the 13C phenylalanine, 14C at the time, phenylalanine in livers of pigs decrease until there was a point whereby further increases in the amino acid or the protein produces no further retention in the indicator amino acid. And so this is the protocol we use. We've developed a minimally invasive protocol whereby we use breath samples, we feed the isotope orally, and we collect urine as a surrogate for blood. And so we have a 2-day adaptation of the participant to an adequate diet, providing 1 gram per kilo of protein. And on the oxidation study day, we bring them into the hospital where we provide 8 hourly isonitrogenous isocaloric meals. At the 5th meal, we provide a prime and a continuous infusion of the stable isotope, usually C13-phenylalanine and C13 labeled bicarbonate, and we provide that in the subsequent hourly meals. We collect breath and urine samples at baseline before the isotope and subsequently at 2.5 hours after. We also measure VCO2, the production of carbon dioxide, in order to calculate the rate of production of 13CO2, which is our main endpoint. And so we analyze our samples, our breath samples on isotope ratio mass spec and our urine samples on tandem mass spec. And so using this method, we've studied the protein requirement in young adults. And prior to studying the protein requirement, we actually reanalyzed the world's nitrogen balance data, the set of data that was analyzed by Rand et al., but we included in that analysis studies that incorporated higher intakes of protein. And so applying not a linear regression, but a 2-phase linear regression analysis to the method, we found a different breakpoint than that which was found by Rand et al., and I'll tell you what that was. This was 0.91 grams per kilo per day as the mean and 0.91 and 0.99 as the RDA. And this is different than 0.6 and 0.8. And so applying the IAAO method to study the protein requirement in young adults, we found a breakpoint of 0.93 and a RDA of 1.24 milligrams per kilo per day. We applied the same method to the study of protein requirements in older adult males and older adult females. And what we found was that the requirement was 0.96 and 1.29 for males and 0.94 and 1. For females and 0.94 and 1.24 for males. And so just to summarize, the DRI recommendations is 0.6 and 0.8 grams per kilo per day. And this is for old and young adults. We reanalyzed the nitrogen balance data and found 0.91 and 0.99. And we applied the IAAO, stable isotope-based IAAO method to study the requirement in young adult males, in older adult males, and older adult females. And we got around 0.9 and 1.2, 1.3 grams per kilo per day. And the requirement in young and older adults, as you can see, these requirements are not different. Now, we were criticized that our estimate of the protein requirement using the IAAO method is too high. And so we applied some stable isotopes. We use an N+3 glycine to assess glutathione kinetics. We looked at fractional and absolute synthesis rates of glutathione, which is a tripeptide, and it's made up of glycine, cysteine, and glutamate. And cysteine is the rate-limiting amino acid for glutathione synthesis. And so we did this study because prior studies from Vernon Young's lab and Jackson's lab had shown that glutathione is sensitive to changes in protein intake as well as sensitive to changes in amino acid intake. And Shekhar et al. In 2011 had shown that when you supplement older adults with cysteine or glycine, there was an increase in glutathione synthesis. And so our question was, what is the level of protein intake to maximize glutathione synthesis in young and older adults? And so we used the precursor product method and an intravenous infusion of an N+3 glycine. We provided 10 hourly meals to the participants. We obtained blood samples for baseline before and a number of samples after. And so this method was a repeated measures design where we used 6 different intakes of protein and measured the glutathione synthesis rates over time. And as you can see, we use a prime continuous infusion of an N+3 glycine. We use tandem mass spec to analyze the concentration and enrichments of glutathione. And also, because it's a precursor product using steady state kinetics, we analyzed to ensure that we had steady state of glycine in the precursor pool. And then we calculated fractional and absolute synthesis rates using these equations. Now, this was published. This is one of my PhD students who did this work in young adults. And what we found was that the amount of protein that maximized glutathione synthesis was 1 gram per kilo per day. And we also analyzed sulfates in these subjects as well. And you can see glutathione erythrocyte absolute synthesis rate increased to a certain point as well, but the fractional synthesis rate was maximized at 1 gram per kilo per day. We also did the study in older adults, and we also found that the amount of protein that optimized glutathione synthesis rates was about 1.1 gram per kilo per day. What was interesting is that if we had used concentration alone in these studies and not applied the stable isotope to measure the kinetics, we would have seen no difference, because from this graph, you can see that glutathione concentration did not change in the older adults, did not change in the younger adults. But what we saw was an increase in glutathione synthesis when we used stable isotopes to a break point where further increases in protein beyond 1 gram per kilo per day did not produce further increases in glutathione. And as you can see, urinary sulfate excretion increased as we increased the intake of protein beyond about 1 gram per kilo per day. And so just to summarize again this data, the DRI for protein is 0.6 grams and 0.8 grams per kilo per day. Reanalysis of the nitrogen balance, about 1 gram per kilo per day. Protein requirement in young and older adults, about 1 gram per kilo per day. Using glutathione as a measure of adequacy or the amount of protein that is required, it's we showed about 1 gram per kilo per day. And so we now was at a crossroad, recognizing that protein requirement is not different between young and older adults, but there's so many physiological studies from Stu Phillips, from Elena Volpe, from Robert Wolfe, from Douglas Patton Jones. There's so many studies that should suggest anabolic resistance, and there's an increased need for protein in older adults and or leucine. And so we wanted to look at amino acid requirement in older adults to understand what is different between young and older adults. And what we found was that the leucine requirement was over twice time higher for older adults than older adults and than younger adults. Sorry, what we found, the current DRI for leucine is 34 milligrams per kilo per day. In young and old adults, the requirement is 79 with an RD of 81 milligrams per kilo per day. And when we analyze or calculate based on lean body mass, it's 128 in older adults and 115 milligrams per kilogram lean body mass in older males. We also looked at the requirement for the sulfur amino acid because sulfur amino acid is seemed to be limiting in the diets of older adults. And what we found again was that the sulfur amino acid requirement in older males is almost twice that of the current recommendation, and it's about 20% higher for older females. We also looked at the lysine requirement, and lysine is another amino acid that has been studied quite a bit. And as you can see from this table, the leucine requirements of younger adults and the current recommendation is 34, but it's 78, close to 80 for older adults. The sulfur amino acid is 15, it's 26 in older males, 17 in older females, and the lysine requirement is 31, but in older adults, it's much higher, especially in the older females. And so we have applied the indicator oxidation method to study protein quality as well, because there is recent recommendations for the world to move to a more plant-based diets. And so we can use the linear portion of the oxidation curve in order to study protein quality, because indicator oxidation is the inverse of protein synthesis. And so we have applied this method to the study of protein quality. We started with looking at rice, lysine in rice, because lysine is a limiting amino acid in cereal grains. And what we found was that if we cooked white rice and we cooked it really soft without burning, then the availability of lysine in rice was pretty high, about 97%. But if we oven browned the rice, then the availability of lysine was only 70%. We applied the IAAO method using the stable isotope C13 phenylalanine again to study lysine and tryptophan in corn, because corn is consumed by many developing nations quite a bit, and lysine is the first limiting amino acid in corn. Tryptophan is the second limiting amino acid, and what we found was that lysine availability was 71%, tryptophan availability was 80%. So looking at the concept of protein complementation, which we know is important for plant-based diet, we studied sorghum and millet. We studied lysine availability in these grains, and then we complemented these grains with lentils. And after complementation, we saw that oxidation of the indicator decreased 20, almost 30%. And this demonstrated that when we combine a grain and a pulse, then protein quality is improved. Same thing, we studied the methionine, which is limiting again in pulses. We studied methionine availability in lentils and chickpeas, and it was 60, 69, 63%. But when we complemented with rice, the protein quality improved. We saw the oxidation decreased. And so just to summarize, using stable isotope tracers, we demonstrated that nitrogen balance estimates of protein requirements are too low for young and older adults. We saw that glutathione as a physiological outcome measure of protein adequacy agrees with the IAO-derived estimates. we saw that protein requirements is not different between young and older adults, but amino acid requirements for older adults are higher for key amino acids than for younger adults. And so what we have concluded from using stable isotope tracers is that protein quality is of greater relevance in older versus younger adults. And so this has very important implications for planning and assessment of plant-based diets. And so this is my acknowledgements. The research is funded by the Canadian Institute of Health Research, and these are some of the students who have done the work.
Thank you, Glenda, for that nice overview and application into your IAAO method approach. We have time for one quick question, and there's one from the audience concerning your software with isotopic enrichment. Could you advise what user-friendly software is best for calculating 13C enrichment?
Um, I can't, it's gone from my mind now. I don't remember the software we use for the standard mass spec, but I can send that out. It's gone from my mind, actually. I don't remember the software. It's been upgraded so many times. I can send that I can send that out to whoever
Perfect.
wanted
Yeah,
to
if you can
know.
address that after the fact,
Yeah,
no
I
problem.
will.
We appreciate your time and thanks again for your presentation.
Welcome.
We'll
Thank
move
you.
on now to the third speaker